Chapter 19, Blood. So as we start our discussion on blood, we're going to examine the functions and properties of blood. Now, before we get too crazy with some details, I want to alleviate any misconceptions about blood or fallacies that we've been told in middle school, high school, or even college, which is one, the color of blood. So in the past, a lot of people have said that blood is red when oxygenated and blue when deoxygenated. This is unanimously and overwhelmingly false. Blood is red. It's always red. It doesn't matter if it's oxygenated or deoxygenated, it's red. It is brick red when deoxygenated and bright red when oxygenated. So, misspell any mistruths we've been told. Now, as far as its functions and properties, if you recall back from our AP1 lecture, that blood is a liquid connective tissue. So the majority of it is plasma, which is made up of water, but it is consisting of cells, which is surrounded by a liquid matrix, again, plasma. So we look at the breakdown and we see that if we were to take a vial of blood and centrifuge it or spin it down, we get a couple of different layers. We get a formed element layer, which is our red blood cell layer, and this constitutes about 45% of this entire tube. Okay, then we get our blood plasma. Again, this water layer makes up another 55%. And there's other things in there, some plasma proteins, some electrolytes, things like that. We'll elaborate that a little later on in this lecture. And then we get this little white layer, this buffy coat. And the buffy coat is composed of all of our white blood cells and our platelets. Now, the cellular components of our formed element layer, our blood, include red blood cells, also known as erythrocytes, our white blood cells, also known as leukocytes, and our platelets, also known as thrombocytes. So we're going to write this in erythrocytes, our leukocytes for white blood cells, and our thrombocytes for platelets. Now, the plasma portion, as we said, is more than 90% water. We find some plasma proteins in there and other solutes. And again, we're going to elaborate on this later on in this lecture. So let's look at a table to break down whole blood, right? What is made up in our blood. So whole blood, 8% is going to be made up of our blood plasma. So there's that 55% breakdown with our formed elements, 45% breakdown. Let's look at plasma. We get 90 to 92% water, right? Water. We get some other solutes. There's our electrolytes, our dissolved gases, CO2 and oxygen, some nutrients, regulatory substance, and waste products, okay? After that, we look at our proteins, and these are going to be important. We have all our plasma proteins. We find albumins, globulins, fibrinogens. And albumin is going to be 54%, globulins 38%. And these are immune response cells, and fibrinogens. We can move on down to our formed elements. So in that 45%, we see anywhere from 150,000 to 400,000 platelets. Our white blood cell count can range from 5,000 to 10,000. This is a normal white blood cell count. Those include neutrophils, which is the most predominant white blood cell found in circulation, lymphocytes, monocytes, eosinophils, and the least numerous are basophils. Then we have our red blood cells, these erythrocytes. Now, these make up the majority of the formed elements, anywhere from 4.8 to 5.4 million cells in circulation. So let's look at this big bar because this confuses people. We talk about our entire body weight. When I said that 8% of whole blood, that's in comparison, what percentage of all of our body fluids, all the other fluids in and around our tissues, how much blood comprises? So that's 8%. 92% is what makes up our other fluids and tissues. So we can look at a scanning electron micrograph or even a histological view of a blood smear. This is something you'll do in lab to understand what and how these cells look different. So if we look up at a SEM, our scanning electron micrograph, we have a white blood cell, a platelet, here purple, and a biconcave disc, an erythrocyte, a red blood cell. Now again, we look down to the right. Your job in lab, once you look at this slide, is to differentiate the different types of cells, different types of white blood cells. So within a week or so after you complete that lab, you should be able to look here and say, okay, all these red cells right here, these biconcave discs, are red blood cells or erythrocytes. All these little cellular fragments here are platelets or thrombocytes. All this white space, it's not just white space, it is the liquid component of blood. It is plasma. Okay? And then we have our various white blood cells. We have a polymorphonucleated granulocyte. So this is a neutrophil right here. We have, again, another polymorphonucleated, 3 to 5 lobes. That's a neutrophil, another neutrophil. And then we have an agranulocyte here. And again, don't worry about terminology. We will elaborate on this. But we see a nice horseshoe-shaped nucleus, right, with an agranular cytoplasm. So this guy right here is a monocyte. So that's a pretty good representation of a monocyte. Functions of blood. Now, blood is going to serve multiple functions, and I highlighted in red here some of those major ones. Number one, transport. So we've already done endocrine, so we know that it definitely transports hormones, right? We know that it transports oxygen and carbon dioxide. Now you can elaborate on this. We can talk about loading and unloading oxygen and carbon dioxide. So think about the lungs. What's happening there? We are loading up on oxygen and unloading carbon dioxide. Now we flip it to the tissue level. What happens there? It's the reverse, right? We're unloading oxygen and loading up on carbon dioxide. So we get this perpetual cycle of loading and unloading. We're going to help transport nutrients to our tissues. We already said hormones. Heat. Think about exercising. Think about when it's cold out. Your fingers turn white. Why? We are redistributing blood to the core, to our extremities, to conserve heat or release heat, which is why our skin turns red when we exercise. And we're also going to transport waste products. Where? To the kidneys for removal, to and through the digestive system, etc. So, we're also going to see that blood regulates. What's it regulating? It's maintaining homeostasis of all of our body fluids. It's regulating pH, acid-base balance, body temperature, and water content or osmolarity within our cells. It's going to protect, which is our third and last function. It protects against excessive blood loss by forming clots. Those thrombocytes, those platelets that we saw are going to form clots against preventing blood loss. And it's going to set up our immune system. So those white blood cells are going to be there to protect us against foreign invaders and infections, any type of pathogen that aims to do us harm. Table 19.1 is going to elaborate on some of the substances in the blood. So let's look at this, and we're going to talk about it in a little detail. So water, that roughly 92% of the plasma is the liquid portion of our blood. It's a solvent and suspending medium. We're suspending all of our other formed elements and proteins and whatnot. And it's going to absorb, transport, and release heat. Our plasma proteins, key note, this is 7%. Most of them are made right here at the liver. Their responsibility is to maintain colloid osmotic pressure. This is what's contributing to our blood viscosity. It will also set up pressure differentials for moving things in and out of our capillary beds. We're going to transport hormones, these are steroids, fatty acids, and calcium, and also help regulate blood pH. Now the smallest and most numerous, and we'll highlight it for you, the smallest and most numerous plasma proteins are our albumins. Now these albumins help maintain osmotic pressure, and they're really, really important in the exchange of fluids across capillary walls. The second are globulins. These are a relatively large protein. They are the cells that produce immunoglobulins. So these are part of our immune system. These immunoglobulins are going to attack viruses and bacteria, and their alpha and beta globulins transport iron, lipids, and fat-soluble vitamins. The last protein we see is fibrinogen. Now this is a relatively, again, large protein, and this protein plays a pivotal role in blood clotting. Without fibrinogen, we wouldn't be able to form blood clots. Next, we look at that 1.5% other solutes that's dissolved in plasma. Electrolytes. These include inorganic salts, positively charged cations like sodium, potassium, calcium, magnesium. We get some negatively charged anions, chloride, hydrogen phosphate, sulfate, and bicarbonate. Now, bicarbonate is going to be an important one, and we're going to relook at this one. And again, this is one of our great buffers of our body. Some nutrients that we're going to look at are products of digestion, such as amino acids, sugars, glucose, fatty acids, glycerol, vitamins, and minerals. These are essential roles in cell function, growth, and cell development. Gases, we talked about dissolved gases, oxygen, carbon dioxide, and nitrogen. So the carbon dioxide is key. Remember we said that blood is going to help regulate pH? Well, here it is. CO2 is involved in the regulation of blood pH. Regulatory substances, we deal with enzymes, hormones, and vitamins. Those enzymes are going to help catalyze chemical reactions. Hormones regulate metabolism, growth, and development. Vitamins are cofactors for enzymatic reactions. And then lastly, we have our waste products. Urea, uric acid, creatine, creatinine, bilirubin, and ammonia. Now, these urea and uric acid, most of these are byproducts. They're byproducts of protein metabolism that's moved through the blood and going to eventually work its way to either the feces or through the urinary system for excretion. The next question we have to answer is, where and how do we make these blood cells? So lymphocytes are able to live for years, while most other blood cells live for hours, days, or even weeks. It varies. The number of red blood cells and platelets remains rather steady, while that of white blood cells varies depending on invading pathogens and other foreign antigens. Now we talk about the process of making blood cells. It's called hematopoiesis or hemopoiesis, literally creation of red blood cells. Now, pluripotent stem cells differentiate. So here's back to our word of classification of all living things, differentiation. So we start with one singular stem cell, a pluripotent stem cell that will be signaled to differentiate into each of the different types of blood cells. Now that includes red blood cells and white blood cells. We can use this flow chart to look at the origin and development of blood cells. Now, I'm going to talk about the colony forming units, the CFUs, but you're not responsible for knowing this. What you are responsible for knowing is the lineage, this phylogeny of what happens and what do they become. So let's outline this. Let's look at our pluripotent stem cell. It's going to give rise to two types of cells, a myeloid stem cell and a lymphoid stem cell. Now, what happens? Let's take that myeloid stem cell. That myeloid stem cell, we're going to box out everything from here to here. Okay? The job of that myeloid stem cell is to turn into, depending on the signaling. So the signaling would be the CFUE, the colony forming unit erythrocyte, which would signal that myeloid stem cell to become a pro-erythroblast. From there, it will go through the varying stages of a red blood cell to form a reticulocyte by about day 7. So around right here, day 7. So at day 7, we have this reticulocyte being formed. Notice it is a nucleated cell. The nucleus is ejected at day 7. It's kicked into circulation, and at day 10, it becomes a fully mature erythrocyte, a red blood cell. So the time frame, how long does it take to make a circulating functional? That's the key, because day 7 is circulating, but it's not till about day 10 that it is a functional red blood cell. So the answer is 10 days. We're also going to see colony-forming unit megakaryocyte. Megakaryo means large body site cell. This myeloid stem cell will lead into a megakaryoblast. So again, blast meaning creation of a megakaryocyte. And it becomes this large bodied cell, megakaryocyte. Now, what happens? The analogy I use is throwing a rock through a plate glass window, and that window shatters into hundreds of thousands of pieces. Well, that's exactly what happens with this megakaryocyte. It fragments into our platelets or thrombocytes. Now, our colony-forming unit granulocyte, this is going to make all of our granulocytes. Okay, it's going to make our eosinophil, so our eosinophilic myeloblast is going to turn into an eosinophil, and we'll get into numbers and functions in a little bit. Our basophilic myeloblast will give us our basophils. Our myeloblast will give us our neutrophils, and then we turn into the first granulocyte we see, which is a monocyte. Now, that takes care of the myeloid stem cell. Now, our lymphoid stem cell, we're going to flip over to this area right here. Our lymphoid stem cell has its own phylogeny because there's so many different lymphocytes, from natural killer cells to plasma B cells to T cells and cytotoxic T cells to helper T cells, CD4 or CD8 cells they're also called. We look at memory T cells, regulatory T cells, memory B cells. So this wide array of lymphocytes has to come from its own lineage, and that's where this lymphoid stem cell. And it gives rise to T lymphoblast, which will give us our T lymphocytes. Our B lymphoblast gives us our beta B cells, and our NK lymphoblast gives us our natural killer cells. So we'll talk about what these guys do, but real quickly, natural killer cells, these guys are really hunting and killing cancer in the body. Plasma B cells are antibody creating cells, so they create antibodies creating. And cytotoxic T are hunter. They are antigen specific hunter cells, so they hunt a specific antigen presented on a infected cell. Let's hone on in and focus on to our red blood cells, and we're going to elaborate its function, its structure, what it looks like, how we identify it. So red blood cells, remember we also call them erythrocytes, contain a protein. They are little sacs of protein. That protein is hemoglobin. Now that hemoglobin is used to carry oxygen to all the cells in the body and to carry some carbon dioxide to the lungs. Each hemoglobin molecule contains an iron, this iron ion, which allows each molecule to bind 4 oxygen molecules. Now this is interesting. Now we say that red blood cells have hemoglobin that has 4 subunits. Each subunit has a iron group in it, and those irons are binding oxygen. So each hemoglobin carries 4 molecules of oxygen. Now we never really elaborate in middle school, high school, or even college about these red blood cells. So we assume that because hemoglobin binds 4 oxygens, that each red blood cell carries 4 oxygens. So we need to put this to reality. And this is going to be important, especially when we're looking at pathologies and diseases and protozoans that take advantage of the knowledge of what's going on in these red blood cells. So remember, I just said that red blood cells are essentially satchels, pouches, bags of protein. So we are no longer seeing it as one molecule of hemoglobin. Each red blood cell, just to put this to real life numbers to blow your mind, each red blood cell carries, you ready for this? Roughly 300 million hemoglobin molecules. 300 million little proteins. So that means if each one carries 4, we're over about 1 billion molecules of oxygen carried in each red blood cells. That's amazing. That's a number that you really have to wrap your head around, okay? So just put some real numbers to what this does. Now, remember, our red blood cell is a biconcave disc. It has no nucleus or other organelles. So no mitochondria, no Golgi. It does have a nucleus at one point during its phylogeny, during its development. We discussed that. And then remember, day 7, when it becomes a reticulocyte, it kicks out that nucleus. But now it's essentially just a bag. And by becoming just a bag, that means that it allows them to carry more oxygen and more protein more efficiently. But now what does this mean? No nucleus means that it cannot undergo mitosis. So we look at these words like mitosis, cell division, and we say, no, it cannot do that. So it has to have a lifespan. How long does a red blood cell live for? This is a question we're going to answer in the next few slides, but I want you to think about that and try to come up with the answer on your own. Let's continue with our red blood cell. So we know that our red blood cell shape is a biconcave disc. It's about 8 microns wide. We look at its protein molecule. So we have our hemoglobin. It's made up of 4 hemes. Each has an iron. And the globin, which is 2 alpha chains, an alpha 1, an alpha 2, a beta 1, and a beta 2. Now, we have to elaborate a little bit here, okay? Now, if we go further on and look at that heme group, you see the heme contained with that iron in the center. So we talk about the destruction of red blood cells and how we recycle them. We're going to allude to organic versus inorganic components. So organic versus inorganic components, okay? So what are the organic components and what are the inorganic components? So the only inorganic component that we're dealing with here is going to be the iron, and that's going to be important when we talk about the digestion and recycling of a red blood cell. So keep that in the back of your mind. So continuing with our red blood cell, hemoglobin is involved in regulating blood flow as well as blood pressure. Now, it regulates blood pressure through the release of nitric oxide. Now, nitric oxide causes vasodilation, so all those blood vessels will open up, wide open, which improves blood flow and enhances oxygen delivery. Now, our red blood cells also contain carbonic anhydrase, an enzyme that's going to catalyze the conversion of carbon dioxide and water to carbonic acid. Now, that carbonic acid transports about 70% of carbon dioxide in the plasma. So a couple things. We cannot carry carbon dioxide in the blood. We always say that we load up oxygen, unload CO2. Carbon dioxide is a gas. You don't want your blood bubbling. Think of a can of cola or soda. You shake it up, you open it up, that carbon dioxide dissociates, and then you get all those violent bubbles. We don't want this in our blood, so we have to convert it. We convert it to carbonic acid, which is done through this enzyme, carbonic anhydrase. Now, the carbonic acid is, again, it's an acid. We don't want to carry this through the blood. So we're going to convert this to bicarbonate, our friendly buffer that we use again and again throughout our body. So we'll look at this conversion later on. But again, we're talking about carrying it as a nice buffer as bicarbonate. Next, we're going to talk about the life cycle of red blood cell. Remember before I said it has no nucleus, so it must have a lifespan. That lifespan is 120 days. So about 4 months the red blood cell lives in circulation. Dead cells are removed from circulation by the spleen and the liver. In our next slide, we'll look at that process. Breakdown products from the red blood cells are recycled and reused. So I inserted this nice little handy dandy slide to help us get through the stages of red blood cell genesis or development. So we start at the stem cell. We have a hemocytoblast that now becomes a committed pro-erythroblast. So that pro-erythroblast, pro-4 erythro red blood cell formation, this is now a committed cell, will not turn or differentiate into anything else. Phase 1, ribosome synthesis. We become an early erythroblast to the beginning of phase 2, which is hemoglobin accumulation, a late erythroblast. We also become a polychromatophilic erythroblast. It starts changing color. The reason why it's changing color is because we are accumulating or adding that hemoglobin. Now we become a normoblast, and notice that a normoblast has a nice typical round shape that we see from an erythrocyte, hence called the normoblast. And around day 7, we get phase 3, which is the ejection of the nucleus. Ejecting that nucleus now becomes a reticulocyte. Now this is where it's going to be housed in the bone marrow for a couple days or kicked into circulation. Now day 10, so we're at 10 days, we are in circulation fully matured as an erythrocyte. Recall that our red blood cell sits in circulation for 120 days. So, what happens? What happens before? What happens after? So we discussed the before, right? We discussed the 10 days it takes to essentially make a red blood cell and puts it into circulation. Our discussion now is what happens after. What happens at 120 days? So we take those red blood cells and we're going to essentially kill them. The red blood cells die, and then we have these macrophages. The macrophages are located in the spleen, the liver, or even in red bone marrow, and they're going to kind of gobble up. They're going to gobble up, like little Pac-Man, gobble up this red blood cell. And they're going to gobble it up and break it into some of its smaller components. It's going to break it into globin and our heme group. Now, all these molecules of the globin are going to get broken down into their subsets, into amino acids. And those amino acids are then going to get kicked out of the macrophage and they're going to be reused for protein synthesis. So that's what we do with the protein portion, right? That organic portion. Now we take that heme group. And we're going to take the heme, and because there's iron there, right, we're going to break it down. We're going to separate the iron, the inorganic, from the organic. And that byproduct is going to be biliverdin. So we'll get back to biliverdin. Let's look at this iron. So the iron, the inorganic portion, is going to get kicked out. It's something we can't digest, we can't dissolve. So we're going to need a transport protein, because again, it can't transport freely in the body. Transferrin is going to take that iron, going to bring it to the liver, at which point the liver is going to kick it out. The liver is going to, again, once kicking that iron out, transferrin is going to bind to it. Now, where's it going to go? That protein is going to bring it right back to the bones. Now, why is that important? Master recyclers, that's what our body is. We're recycling this inorganic portion, the part we can't break down. So we take that iron we just recycled, we mix it with our globin protein again, with vitamin B12, and erythropoietin is that EPO, that hormone that's going to stimulate red blood cell production, is going to kick off erythropoiesis in the red bone marrow. And then here we go, back at the start, 10 days, we get red blood cell in circulation. Well, we still have this waste product, biliverdin. Biliverdin is going to get converted to bilirubin. Now, again, we're in the liver. The liver is going to kick it into the small intestine, so that hepatic portal system. And then the small intestine is going to break it down further. It's going to break down into bilirubin. So bilirubin with this bacteria digestion, so our bacteria that's in our gut is going to break down that bilirubin into urobilinogen. Now the urobilinogen is going to go two ways. It's going to get broken down even further to stercobilin and then eliminated in our feces, and some of it's going to go to the kidneys, and it's going to get broken down even further into urobilin and then going to get excreted in our urinary system, in our urine. We introduced a word on this last slide called erythropoiesis, and we didn't really define it yet, so we're going to define it here. Erythropoiesis is the production of red blood cells. It does begin in the red bone marrow. Now, there is a hormone that's going to be released from the kidneys. It's called erythropoietin, or EPO. Now, we release EPO from the kidneys in response to low oxygen concentrations, or hypoxia. This is going to stimulate differentiation of hematopoietic stem cells into erythrocytes. Now, again, some key words. We have this reticulocyte. This immature red blood cell enters the circulation at about day 7, and in 1 to 3 days, it matures to a full functional erythrocyte. We're going to finish up with red blood cells to look at a feedback loop. So disruption in homeostasis by decreasing a control condition, which is oxygen delivery to the kidneys and other tissue. Those kidney cells detect low oxygen levels, increasing EPO secretion into the blood. This input moves to the control center, where proerythroblasts and red bone marrow matures more quickly into a reticulocyte. The more reticulocytes entering circulating blood, the effectors, large number of red blood cells are now in circulation. The response, we increase oxygen because we're increasing red blood cells. We're increasing oxygen delivery to the tissues. Once we return to a homeostatic condition or a balanced condition, when oxygen delivery to the kidneys increase back to normal, we shut the system down. So it's our negative feedback loop. The next grouping of cells we're going to look at are our white blood cells. Our white blood cells are also called leukocytes. They're a little bit different than red blood cells in that they contain a nucleus and organelles, but they do not contain the protein hemoglobin. Now, we classify our leukocytes and we put them into two categories. They're either granular or agranular, and this alludes to the speckles or the grains found within the cytoplasm. So, granular leukocytes are containing vesicles that appear when the cells are stained. Our granular leukocytes include our basophils, our eosinophils, and our neutrophils. Our agranular contain no granules. These are agranular leukocytes, which include our lymphocytes and our monocytes. White blood cells. We're going to take a look at the histology of these white blood cells. Again, this is something you do in lab with the microscope, finding and identifying these blood cells based on their nucleus, the granules, no granules, color, shape, etc. Again, my tutorial channel on YouTube, Anatomy and Physiology Crack, goes through each and every one of these, but we are going to identify them now. So what we're going to do is we're going to bracket these three and these two. These three are granulocytes, and these two are agranulocytes. So these are our categories, our two categories. Now we look at the most predominant white blood cell in circulation, 60 to 70%. These are our neutrophils. Now, what do we know about our neutrophils? They are, one, polymorphonucleated. What does that mean? That means the nucleus can have anywhere from 3 to 5 lobes. Okay, and this one has 1, 2, 3, 4, 5 lobes. So this is definitely a neutrophil. Now, what else do we see? It is granulated, and color. It is a purplish, reddish, pink. Okay. Now, our next cell, and again, we're going to go into the details as to what they do and their percentages of distribution a little later on in this lecture. So we're just going to use the color, texture, shape, size to identify their histology. Next, eosinophil. Eos means morning dawn. This one is acid loving, which is why we get this staining, the granules stain red. So one red stained granules is the only one that appears this way, definitely eosinophil. 2, look at the nucleus. Looks like bunny ears. It is bilobed. Remember, this is not multiple nuclei. It's one nucleus shaped differently. So a bilobed nuclei, almost like bunny ear. So again, key identification markers, red stained granules, so beautiful red, and bilobed nucleus. Next, basophil. This is the least circulating, less than 1%. This guy has one, also a bilobed nucleus, but because it is so heavily stained, so heavily granulated, we cannot see the nucleus. So these guys are small. These guys really turn into mast cells. They degranulate, releasing things like histamine, serotonin, things like that. So these guys, again, bilobed, but the granulation is so heavy and the stain is so dark you can't really see the nuclei, and they're also relatively small. Now, for agranulocytes, we get our lymphocyte, number 1. It's obviously agranulated, no granules in that cytoplasm. You get some key markers. A sliver of cytoplasm is visible, and that's because the nucleus is so large. So nucleus is large. So look how big that nucleus is, and we're going to zoom on in to make the drawing a little bit easier. And there's our cytoplasm, it's barely surrounding the nucleus. You can barely see it. Now, the biggest mistake people make when they're looking at lymphocytes in in the microscope is that they see that massive nucleus, and sometimes, remember, these cells are turned or rolled, and the sliver of cytoplasm here is even smaller, and people mistake it for basophils. Remember, basophils are less than 1%. You might not even see a basophil in your field of view, or even on your entire slide, you might not even find one. So really phase in and out, look closely at those lymphocytes and see if you see that sliver of cytoplasm. The next here, I call this big foamy. So again, big foamy. These are words, trigger words that I get you to really see what this looks like. So this is one of our larger white blood cells. That nucleus is huge, horseshoe shaped. So horseshoe or kidney shaped. Kidney shaped. I wrote horseshoe, but you can do kidney if you want. And that nucleus is, again, big and foamy. And no granules. So it's an agranulocyte, so it's nice and clear. So that's our key marker. Now, some features about white blood cells: they can live for several months or even years as they set up our immune system. Their main function is to combat invading microbes. Now, during an invasion, many white blood cells are able to leave the bloodstream and collect at the site of invasion. Let's think of this like you get a cut in your skin, the superficial cut. Now, we have bacteria, staph, all over our body. Anytime you get an abrasion, a cut, that staph is trying to get in and do damage to you. It's trying to take hold. Now, luckily, our white blood cells are going to come here, and they're going to leave the bloodstream through this process called emigration. Now, what happens? They physically walk through the endothelial cell lines of the blood, of the capillaries, of the blood vessels, and get into the tissues and work their magic, which is essentially remediating that infection. The process of emigration is called diapedesis, dia meaning through, and ped meaning feet, or like walking, so it can walk through these layers. Our next slide is going to look at a diagram to explain this. So here's a picture explaining the emigration of white blood cells. So blood is flowing down, so we see this right here, and we have our white blood cell. Now, on the surface of these white blood cells are proteins called integrins. Now, these integrins, again, located on the surface of these neutrophils, are going to work with their counter receptors called selectins. So we see these little circular selectins, and basically what they do is that they're going to allow for and contribute to leukocyte and endothelial cell activation. So we're basically signaling trauma, right? And these guys are going to work together to allow for the sticking of the integrins to the cytoskeleton of the endothelial cell line. Once there, the cell will actually squeeze. Once it's stuck, it will start squeezing between those endothelial cells. Let's examine our white blood cells even further from a clinical perspective. Now, what could we do with them? How do we know, one, how many there are? Remember, we said generally in circulation we get anywhere from 5,000 and 10,000. This is a normal white blood cell count without an infection. So in general, when that white blood cell count is greater than, so our white blood cell count is greater than or equal to 10,000, this can indicate an infection. Now, this clinically has a term. The term is leukocytosis. So again, white blood cell count higher than 10,000 is called leukocytosis. Now, what do we do with that? What does this mean? Yes, it tells us we have an infection or inflammation. Now, we can use a differential white blood cell count to tell us specifically which white blood cells are more predominant. For example, if you have eosinophils, so if you have a high count of eosinophils, this can indicate a parasitic worm infection, right? So the differential white blood cell count will help us determine what the problem is specifically based on that specific white blood cell. Now, on the flip side, let's look at a white blood cell count that is less than 4,000 white blood cell count. This is called leukopenia. Now, what are the causes of leukopenia? So low white blood cell count could be due to things like acute or severe viral infections, things like the cold or influenza. This can bring your white blood cell count less than 4,000. It's also been associated with things like chemotherapy, where we knock out the immune system and reduce the number of available white blood cells. Radiation therapy, myelofibrosis, aplastic anemia, or other conditions. Also, stem cell transplant. Again, remember when we're doing stem cell transplant or bone marrow transplant or even organ transplant, we knock out the immune system, so we reduce the chance of rejection. Also, any type of immunocompromised disease, HIV, AIDS, and also steroid use can also lower your white blood cell count. Table 19.2 goes through essentially a differential white blood cell count. So what these high or low count can indicate due to infection or due to pathology. So let's look at our neutrophil. A high count can indicate bacterial infection, burn, stress, or inflammation. A low count indicates radiation exposure, drug toxicity, so certain drugs you take for infections or for disease can lower your white blood cell count. Vitamin B12 deficiency or systemic lupus. Now, lymphocytes, high count, viral infections, some leukemias, and infectious mononucleosis. Low count is prolonged illnesses like HIV infections or any type of immunosuppression or even treatment with cortisol. Look at our monocytes. Remember big foamy? Well, our high count can indicate viral or fungal infections, things like tuberculosis, some leukemias, and other chronic diseases. Low cell count equals bone marrow suppression treatment or treatment with cortisol. Eosinophils. We see high count, allergic reactions, parasitic worm infections, and autoimmune diseases. Low count indicates drug toxicity, stress, acute allergic reactions. Basophils, again, are mast cells or degranulators. These are allergic reactions, some leukemias, cancers, or hypothyroidism. Low count could be indications of pregnancy, ovulation, stress, or hypothyroidism. The next type of cells that we find in our blood are platelets. So these are derived from megakaryocytes. So they're found in red bone marrow that splinter, shatter. Remember our plate glass window analogy? So that big cell body, that megakaryocyte, splinters into 2 to 3,000 fragments, creating these platelets. They contain many vesicles, but no nucleus, no nuclei. Platelets are going to be used for clotting, so hemostasis, so stopping bleeding or blood loss. Under the influence of certain hormones like thrombopoietin or hemopoietic stem cells are going to differentiate into our platelets. Now, the lifespan of our platelets is much less than our red blood cells. They can survive for only 5 to 9 days. Now, table 19.3 summarizes all our formed elements, like red blood cells, all our different white blood cells, the numbers that we find in males and females, their characteristics, and their functions. So kind of summarizing everything that we have talked about regarding blood. We continue with that table to look at our agranular white blood cells and our platelets. Stem cell transplants from bone marrow and cord blood. So two real important therapies. So bone marrow consists of hematopoietic stem cells, and these are stem cells that can transform into any one types of the red blood cells or any of the white blood cells. So we can use this knowledge to harvest bone marrow from the iliac crest of healthy individuals and match donors to replace cancerous bone marrow in people suffering from cancer. And this is, again, a really good therapy to help treat people suffering from bone marrow cancer. Now, a second way that we can find even more rich stem cells is to collect them from frozen or harvested umbilical cords that have been collected after birth. Now, these have a bigger advantage over basic bone marrow transplants because the stem cells found in umbilical cords are rich in not only hemopoietic cells, but also mesenchymal cells. So these cells could not only differentiate into any one of the white blood cells or even red blood cells, but they could also differentiate into any type of cells, tissues, nerve, and even muscle tissue in the body. So the implications of utilizing these stem cells reaches far and wide. We can theoretically collect stem cells from a baby, and you can use those undifferentiated cells to really treat disease across your entire familial line. So there are companies now that are offering technology to store, it's a paid for service, store your umbilical cord so that you can use it further down the road. Again, anybody in your family that is a genetic match to you can utilize these stem cells to treat disease, cancer. The list goes on and on. Again, once the technology is available, but it's kind of a bet on the future. The next topic we're going to talk about is hemostasis. So hemostasis is a sequence of responses that's essentially going to stop bleeding, and we can term it the clotting response, right? So the process involves a couple steps. 1, vascular spasming, 2, a platelet plug formation, and 3, the coagulation or blood clotting phase. Platelet plug formation. Before we get into the actual platelet plug, we have to understand what's going on in these blood vessels that prevent actual clotting when there's no injury. So the endothelial cells of intact vessels, they do prevent clotting by expressing a fibrinolytic heparin molecule and thrombomodulin. Now, what these two do is they prevent platelet aggregation and stops the coagulation cascade by using and utilizing nitric oxide. Now remember, nitric oxide keeps these blood vessels wide open. Now when endothelial injury occurs, the endothelial cells stop secretion of coagulation and aggregation inhibitors and instead secrete one of our factors, one of our clotting factors called von Willebrand factor. Now what this does is it causes platelet adherence during the initial formation of a clot. The vasoconstriction that occurs during hemostasis is a brief reflexive contraction that causes a decrease in blood flow to the area. We call this vascular spasming. Now, the platelets create the platelet plug that forms almost directly after a blood vessel has been ruptured. Now, it takes about 20 seconds of an injury, within 20 seconds of that injury, in which the blood vessel's epithelial wall is disrupted, coagulation is then initiated. It takes another 60 seconds until the first fibrin strands begin to intersperse among the wound. After several minutes, the platelet plug is completely formed by fibrin. Now, contrary to popular belief, people believe that clotting of skin, especially skin injury, is caused by exposure to air. This is actually not the case, but by the platelets adhering to and being activated by collagen in the blood vessel's endothelium. Now, the activated platelets then release the contents of the granules, which contain a variety of substances that stimulate further platelet activation and enhance the hemostatic process. Now, basically what happens is this. The lining of a blood vessel breaks, the endothelial cells are damaged, and what happens is this reveals the subendothelial collagen proteins from that extracellular matrix. Now, thromboxane is released, which causes platelets to swell, grow filaments, and start clumping together or aggregating. Now again, that von Willebrand factor causes them to adhere to each other and then the walls of the vessel. This continues as more platelets congregate and undergo the same transformation. The result is this platelet plug that we see down here. Now the coagulation cascade is a little bit different. If the platelet plug is not enough to stop the bleeding, the third stage of hemostasis begins, the formation of a blood clot. The platelets contain secretory granules. When they stick to the proteins in the vessel wall, they tend to degranulate, and then as a result, they release their products, which includes ADP, serotonin, and thromboxane A2, which we see right here. And these things are going to activate other platelets. Now what happens first is blood changes from a liquid to a gel. Well, we deal with our 12 coagulating or clotting proteins or factors that are going to act in and outside, so intrinsically and extrinsically to the wound. And we'll talk about those pathways in a little bit. Now, each of the clotting factors, again 12, have a very specific function. We have prothrombin, thrombin, and fibrinogen are going to be the main factors involved in the outcome of the coagulation cascade. Now remember those. Prothrombin again, thrombin, and fibrinogen. Now, prothrombin and fibrinogen are proteins that are produced and deposited in the blood primarily by our liver. So remember we said that the liver is going to make a majority of our plasma proteins. Well, here we are. Here's fibrinogen. Now, when blood vessels are damaged, vessels and nearby platelets are stimulated to release a substance called prothrombin activator, which is going to actually activate the conversion of prothrombin, a plasma protein again, into an enzyme called thrombin. The reaction does require calcium ions. Now, thrombin is going to facilitate the conversion of a soluble plasma protein called fibrinogen into these long, insoluble fibers or threads of the protein called fibrin. Now, these fibrin threads wind around the platelet plug at the damaged area of the blood vessel, forming an interlocking network of fibers and a framework for the clot. Now, what this is going to do is essentially it's like Spider-Man who spins a web and slings it against the wall. Now, this spider web or net is going to trap and help hold the platelet, blood cells, and other molecules tight to the site of injury. And initially, this functions as the initial clot. This temporary fibrin clot can form in less than a minute and is going to end up slowing blood flow before platelets attach. Now, we're going to get into more of the details of the different steps and the pathways in a little bit, but what we wanted to cover right here was what happens with the site of injury and how those collagen fibers and damaged endothelial cells signal this cascade right here. And then again, formation of that fibrin Spider-Man spiderwebby connection that's going to form that platelet plug. So blood clotting involves several clotting or coagulation factors or proteins. They're numbered 1 through 13. Now again, in the last slide we said 12. Well, yes, there are 12, but we numbered them 1 through 16, but 5 and 6 have since been deemed the same clotting factor. So even though they're numbered 1 through 13, we are missing 5 and have traditionally 12. Now, blood clotting is going to be activated by one of two pathways, the extrinsic pathway and the intrinsic pathway. Now, the intrinsic pathway is activated through exposed endothelial collagen, so this is within that damaged blood vessel, and the extrinsic pathway is activated through tissue factors released by endothelial cells after external damage. So again, one outside the body tissue level response versus inside the body of blood vessel response. Now, this pathway is the longer pathway of secondary hemostasis. Now, we talked about primary hemostasis before, and if that doesn't really resolve the blood loss, we enter into the secondary hemostasis now. So, both of these pathways lead to the formation of prothrombinase, and from there, the common pathway continues. So, what we're going to do is look at these two pathways, see which one is longer, and subsequently which one is shorter and which one is deemed more important than the other. So another slide to basically review everything. Extrinsic pathway is our tissue response. So basically the extrinsic pathway begins outside the bloodstream. Our intrinsic pathway is a platelet response, so this is inside or within the bloodstream. We have our 12 procoagulants or 12 proteins that activate each other in a cascade-like fashion. Now, the interesting thing is that this extrinsic and intrinsic pathway are going to converge. It's going to converge onto the common pathway. Now, this common pathway is going to conclude with fibrinogen being converted into fibrin, this insoluble protein meshy network that will cover that platelet plug. So let's take a look at these pathways. The coagulation phase of hemostasis. So we've introduced these three pathways. The extrinsic pathway, again, outside of the blood at the tissue level, and the intrinsic pathway, which is the inside, so the pathway that occurs inside the bloodstream, and then how they both converge onto this common pathway. So let's start with the quicker responding extrinsic pathway. It is a more direct pathway. It begins when damage occurs to the surrounding tissues. Now, this could be a cut like a knife or a stab, so a deep damage, a traumatic injury to a superficial layer. Now, upon contact with blood plasma, the damaged extravascular cells, which are extrinsic to the bloodstream, release this factor, factor 3. It's also known as thromboplastin. Now, following that release, calcium and then factor 7, which is called proconvertin, which is activated by factor 3, are added, forming an enzyme complex. Now, this enzyme complex leads to the activation of factor 10. Now, factor 10 is also called Stewart-Prower factor. Now, the activation of factor 10 is going to activate the common pathway that we're going to talk about in a little bit. Now, as far as time, there's only a few factors here. There's only 3 different factors here. Now, that's important because the events of the extrinsic pathway are completed really, really quickly, and actually in a matter of seconds. The next pathway is called the intrinsic pathway, and again, this is also known as the contact activation pathway. Now, notice that we're dealing with a lot more clotting factors, a lot more clotting proteins. Now, this is important because that means that this pathway is going to be longer and more complex. The factors involved are intrinsic, which means present within the bloodstream. So the pathways can be prompted by damage to the tissues resulting from these internal factors such as things like arterial disease. Now, it's important because it's most often initiated when factor 12, called Hagman factor, comes into contact, and we see factor 12 right here. So let's go ahead and underline factor 12. We'll use black, factor 12. So when factor 12 comes into contact with foreign materials, such as when a blood sample is put into a glass test tube. Now, within the body, factor 12 is typically activated when it encounters negatively charged molecules. Now, these things are, an example would be like inorganic polymers and phosphates produced earlier in the series of the intrinsic pathway reactions. Now, factor 12 really sets off a series of reactions that in turn is going to activate factor 11, which is called antihemolytic factor, factor C, or plasma thromboplastin antecedent. So then it's going to activate factor 9, which is an antihemolytic factor B, or plasma thromboplasmin. Now, while all this is happening, chemicals released by the platelets increase the rate of these activation reactions. Finally, factor 8, which is antihemolytic factor A, which is being secreted from the platelets and the endothelial cells, combined with factor 9, which is antihemolytic factor B, or plasma thromboplasmin, to form an enzyme complex that's going to activate factor 10, again, that Stewart-Prower factor, or thrombokinase. Now, again, by activating factor 10, we are now converged back on, or leading back to, the common pathway. Now, let's talk about time. We said that the extrinsic factor occurs in seconds, and that's because it's shorter and it has less factors. Now, the intrinsic pathway is more complex, much more factors, and look at the time. It takes a few minutes to complete. Now that we have discussed the extrinsic and intrinsic pathways and how they converge onto the common pathway, let's examine what exactly happens in the common pathway. Now, again, both the intrinsic and extrinsic pathways lead to the common pathway. Now, the key point here is fibrin is produced to seal off. So let's see fibrin. So this is where fibrin is produced to seal off the vessel. Once factor 10 has been activated by either the intrinsic or extrinsic pathway, this enzyme, prothrombinase, converts factor 2, which is the inactive enzyme prothrombin, so here's our inactive enzyme prothrombin, into this active form. The active enzyme is known as, and we'll highlight it for you, is known as thrombin. Now, we could take a side note, and let's take a look at what would happen if the enzyme thrombin were not normally in this inactive prothrombin form. Now, this is important because if thrombin was always available, clots would form spontaneously, a condition that's not consistent with life, right? So if we were constantly forming clots, we'd be having these pulmonary embolisms, these clots inside our lungs and in our brains, which would eventually kill us. So again, it's not consistent with a healthy life. So let's continue back. So thrombin is now formed. It converts factor 1, which is this insoluble fibrinogen. So here's our fibrinogen, this insoluble fibrinogen, into a soluble fibrin. And that's what's important right here, that soluble fibrin, which are these protein strands. Now, we have one more factor. Factor 13 is going to now stabilize the fibrin clot. So this slide goes through the blood clotting cascade, similarly that we did on the last slide, with a little different perspective, looking at the extrinsic and intrinsic way up here, and how they converge, so way up here, and how they converge onto the common pathway, and those key steps, right? Thrombin, the conversion of fibrinogen into those loose fibrin, and then here's that factor 13 that's going to reinforce and strengthen that clot. So again, a reintroduction, or not a reintroduction, but at least a examination or re-looking at exactly what happens with this cascading effect. Now you can use either the slide before or this slide, whatever works better for you or makes more sense to help explain this clotting cascade. So from a scanning electron microscope view, we can see the actual stages. So early stage, you see the fibrin threads, these loose threads kind of forming, aggregating. We can see the intermediate stage where more red blood cells are starting to get caught and intertwined within there. And then that late stage, right? So what happens? That late stage, we see an increase in number of fibrin. Again, if you think back to that Spider-Man analogy where he shoots that web, catching everything in its path, that's exactly what's happening here. All these erythrocytes are being trapped in those fibrin threads. And what do we build? We build a massive blood clot that is occluding or blocking the damaged area. Table 19.4 goes through all those clotting coagulation factors and what their names are with their associated numbers, as well as their source and pathways of activation. So the key takeaway here, again, you don't have to go crazy with the actual names, but I want you to realize, look at the consistency. Where are they made? Liver, 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 liver. Remember we said the liver is the site for the majority of our plasma proteins? Well, here they are. Our pathway of activation shows us where each numbered factor is contributing to those cascading effects. So a good overall view. So continuing with hemostasis, we have to talk about what happens once the clot forms. So the clot is going to retract, it's going to tighten, which pull the edges of the damaged blood vessel together. Now here's an important note. Vitamin K is a real important substance that is needed for normal clot formation. And the reason why it's so important is because it's used to synthesize 4 of our major clotting factors. Okay, so the next question is, well, once we form this actual clot, how do we break it up? How is it dissolved? So small unwanted clots are usually dissolved by plasmin, which is an enzyme that is part of the fibrinolytic system. So what we're going to do now is we're going to take some time and we're going to talk about fibrinolysis. So the actual breakdown, lysis, of fibers. So the stabilized clot is going to be acted upon by contractile proteins within the platelets. So again, these proteins contract, they're going to pull on the fibrin threads, bringing the edges of the clot more tightly together. Now I have an analogy that I use to help kind of explain what this looks like. If you've ever really loosened your shoelaces to get your shoe on your foot, and then you have to sit there and tighten them one bunch at a time. So that slow process of tightening your shoelaces is a pretty good example of what happens with those fibrin threads to bring those edges of those clot together. The process is also going to wring out of the clot small amounts of fluid called serum. So any excess serum that's in there, it's going to wring it out, kind of like if you were to twist a towel together to wring out any wet water. Now that serum, which is blood plasma, that serum is blood plasma without its clotting factors. Now the aim is to restore normal blood flow. So in order to restore normal blood flow as the vessel heals, the clot has to be removed. This is called fibrinolysis. Now, fibrinolysis is that gradual degradation of the clot. Now, it's a fairly complicated series of reactions that's going to involve, again, factor 12 and protein catabolizing enzymes. Now, during this process, the inactive protein plasminogen is going to be converted into the active, here we go again, plasmin, okay, which is going to gradually break down the fibrin of the clot. Now, additionally, Bradykinin, which is a major vasodilator, is going to be released. And this reverses the effects of serotonin and prostaglandins from the platelets. This allows those smooth muscles in the walls of the vessels to relax, to open up, and slowly to restore the circulation to the blood vessel. Next, we're going to talk about blood groups and blood types. Now, blood is characterized into different blood groups based on the presence or absence of glycoproteins and glycolipid antigens, which are called agglutinogens, on the surface of red blood cells. Now, for humans, we follow this ABO, so we'll write down ABO blood groups. There are 24 blood groups and more than 100 antigens. Now, because these antigens are genetically controlled, blood types vary among different populations. Now, classification is based on antigen labeling, A, B, AB, and O, with O being the absence of these antigens. An additional antigen, Rh, which is present in 85% of humans. Now what we're going to do is we're going to go to our next slide and we're going to use a whiteboard to kind of explain how this works and how it looks on a red blood cell. So we're going to start with the genetics of blood grouping. So let's look at type A blood. Now you have a couple options for type A. You can either be A for the homozygous dominant trait, or the heterozygote AO. Now, type B blood. You have a similar scenario to A where you have the homozygous dominant BB or the heterozygote BO. The next type of blood we see is type O. Now there's only one way you're going to get type O, and that is the homozygous recessive trait OO. Now the last type we see is type AB. Because both A and B are dominant alleles, they express evenly. This is known as codominance. So the only way you are type AB blood is if you get both the A and the B allele for that trait. Now, an example of this would be a cow. You mate a white cow with a brown cow and you get a cow with white and brown spots. So again, this is called codominance. Now that we understand how it works, we can run a simple scenario to see what our potential blood types would be based on our parents using a Punnett square. So let's say that dad was type A blood and he was the heterozygote AO. And mom was O blood, right? So we know that if she's O, she can only be OO. Now what does this mean? This means you have a 50% chance of being type A and a 50% chance of being type O blood. So again, you can run scenarios all day long. You kind of backtrace your blood type based on, or guess your blood type based on what your parents' blood type is. Now, that takes care of some basic background information on blood type. The next step is to look at how these blood types affect the appearance of our red blood cells. Let's go ahead and start with type A blood and what the red blood cell looks like. So as we can see from our picture, the red blood cell of a person with type A blood has on its surface A antigen. So let's highlight what is presented on its surface, A antigens. Now, circulating in the blood are anti-B antibodies. Now, this is important, okay? These are anti-B antibodies. So, if by accident a doctor infused you with the wrong type of blood, say you're type A and they infuse you with a bag of type B blood, the antigens on the surface of that type B would react to these anti-B antibodies. So these antibodies would stick to all the B antigens on the surface of that B red blood cell and cause these red blood cells to start clumping. Okay, this would be a really bad reaction that you would not want happening inside your body. Let's take a look now at type B. Look on the surface of this type B red blood cell. What do we see? We see B antigens. Look at the shape of those proteins. They are triangular. They fit nicely into these anti-B antibodies. Okay, so that was that reaction that we just got finished talking about. So let's go back to our type B blood and our B red blood cell. So on the surface we get these B antigens, and then in the plasma we get anti-A antibodies. So these anti-A antibodies, we'll highlight them here for you. Anti-A antibodies are going to glutinate or react with these guys right here. Look at the shape of the receptor, rounded. React with A blood, cause a glutination reaction with that A blood. So again, if you have type B blood and somebody infuses you with a bag of type A, you get this glutination where all these anti-A antibodies are going to start clumping and sticking to these red blood cells, causing this glutination. And I'm going to zoom out to put a little animation in to show you what that glutination reaction looks like. And that's going to be important when we talk about our blood typing. The next blood type we're going to look at is type AB. Now let's take a look at the surface of AB. This is an interesting blood type. On the surface, we have two types of proteins, both A and B antigens. Now the interesting note is going to be on the flip side, the antibodies. People with type AB blood circulate no antibodies, so no anti-A and no anti-B. So that means that when it comes to receiving blood, they will not mount a glutination response to either type A or type B or even type O blood. So that makes people with type AB blood known as the universal recipient. So they can receive blood from anyone, type A, type B, and even O. The last blood type we're going to talk about is going to be type O. Type O blood is another unique type of blood. Let's look at the red blood cell. Notice, it is naked, right? I call it a naked red blood cell. It has neither A nor B antigens, proteins on its surface. However, an interesting note is that it circulates both anti-A and anti-B antibodies, which means that people with type O blood cannot receive A, B, or AB blood because their antibodies would mount a response. However, they can donate to whomever. So we can consider them the universal donor. We can look at table 19.5 to examine blood groups and blood types and the distribution of blood types in the United States. So, for example, let's notice that the majority of people in the world, right, and we could even go into the U.S., is going to be type O blood. Native American, close to 80%. European American, African American, we're right on that 50% mark. The next type, let's go, Korean American, Japanese American, Chinese American, we're in that roughly, on average, 36%. The next most predominant blood found in the U.S. is type A. So again, we can see the distribution. European American has the highest percentage, type A, for 40%. Type B blood, the second or third most populous type of blood group in the U.S. And again, European American down 11%, and the majority of people, the African American, Korean American, Japanese American, Chinese American population, an average of about 24-25%. Now the rarest blood type, less than 7% of the population in the world, and even less in the U.S. and even less in the world, is type AB. So those universal recipients, as much as you would love to receive blood from everybody, is the rarest blood type in the U.S. Now, something we haven't talked about is our H factor. which is right here. Now we see that the majority of the population in the US and even in the world has this Rh factor on their red blood cell, this protein on their red blood cell. So we can say European Americans 85%, African Americans 95%, and then Korean, Japanese, Chinese, and Native Americans 100%. And this is a dominant trait. So if you have even one parent who is positive, even if the other is negative, your blood will automatically be positive because of the dominant trait or the characteristics of this Rh factor. Blood groups and blood types continue. Now, blood plasma usually contains antibodies. These are also called agglutinins. They react with A and B antigens. This is what we just got finished talking about on our last slide. Now, an individual will not have agglutinins against his or her own blood type. So again, refer back to our last slide for detailed information on those surface marker proteins as well as the anti-A and B antibodies present in serum and plasma. So our slide here is essentially a review of what I put into our little tutorial on the last few slides, talking about the blood type, what's present on the surface of the red blood cell, and what's circulating in plasma. So again, here, everything put together in one nice slide. Again, you can review back to a few slides ago to see that discussion again. Now, in order to determine someone's blood type, we do a typing and cross-matching. This is what is used typically to, again, determine a person's blood type. This is something we will do in lab. If we are online, then we will work through the steps. Obviously, you won't be able to do this at home, but in a real live class scenario, we would be taking our own blood and running the test that's going to be described in the following slides to decide and determine our blood type. So basically what happens is we take a drop of blood, we mix it with an antiserum. That antiserum has antibodies in it, so anti-A and anti-B antibodies. It will agglutinate or glutinate blood cells, meaning clump them together, that possess those agglutinogens on the surface to react with. So here's an example. The table on the left, the slide on the left right here, is the sample of untreated blood. We then put a drop of blood in well A as well as well B. Now, typically there should be a third well here, which is a D well. This is the RH well, but in this slide it doesn't have it. The samples that we take in lab will have a D well, so we could determine our Rh factor for positive or negative. Now what we do is we put a drop of this anti-A serum in here with the A blood and a drop of anti-B. And what do we see? In the A well, we see glutination, we see clumping. That's what all these little streaks are. We could zoom on in and see all that clumping in there, right? And then in the B well, everything is homogeneous, so there is no reaction. So typically what we say is anytime we get clumping, glutination, we say this is a positive reaction for that blood type. Okay, so that means that this sample here, positive, negative, so positive, negative, so this person is A. We go to our next well, we have our next unknown, homogeneous, and again, clumping. So it's negative for A, positive for B, this person is B. In our next unknown, we're getting both clumping in A, clumping in B. That means this person is positive, positive. So it is AB. In our last unknown blood, homogeneous, homogeneous. That means it is negative for A, negative for B. So this person's blood type is O. Okay? So again, the only difference between this and what we do in lab is that third well type right here, which is anti-D. It is a Rh factor, and same rule goes. If you get clumping, so if there was clumping, say in this third well, you would be A positive. If there was no clumping and it was homogeneous, kind of like this right here, you would be B negative. The next thing we're going to talk about is the clinical significance of Rh factor and what disease can be caused by it. The disease is called hemolytic disease of newborns, or it's also called erythroblastosis fetalis. So we can look at exactly what causes this disease. Now remember we said that Rh factor was a dominant trait. So that means On the rare chance, remember 98% of the population is Rh positive. So on the rare chance that a mother is negative, let's use an example as being O negative, the chances on her significant other being positive is pretty high, which means that when they conceive a child, that child will be Rh positive. Now recall that mother's blood and baby's blood do not mix. They're not a shared circulatory system. Now, so what does that mean for the Rh positive baby and the Rh negative mother? Now, it's inevitable that some of that fetal blood will leak, a little bit will leak into mother's circulatory system, into her cardiovascular system, which means that mother will mount a response. Because she's negative, her anti-Rh antibodies will go set out to mount a response. Now again, during the first pregnancy, it takes some time to build up enough antibodies to do any damage. The first baby is typically okay. The second baby is the problem. So now if you get pregnant again and that baby is positive and again you are negative, that mother is negative, the second time that fetal hemoglobin, that fetal blood leaks into mom's circulatory system, now mom's body says, I recognize this. This is something I've seen before. I have the antibodies, the anti-RH or anti-D antibodies already ready. And it goes transplacental, it goes into the baby and sets out to lyse and destroy all of the red blood cells in that baby. Now, this is a real bad scenario because that baby during development will be starved of oxygen because its red blood cells are being destroyed. The outcome is pretty glim. Now, don't worry because there is a injection that will be given if you are Rh negative. It is called RhoGAM. So RhoGAM is an important injection to get if you are negative. We can use a picture to explain exactly what we discussed on our previous slide. So some of the mom's or fetal blood leaks into mom. You can see those green Rh antigens leaking into the body, and anti-Rh antibodies are now being produced. Second fetus comes, and here they come. Once that leaks back in again, we get some activation of this immune response. And as a result, those anti-Rh antibodies will then set out to attack the fetus. Another disorder we can look at with regards to homeostatic imbalance is sickle cell anemia. Now, it is a genetic condition that causes this sickling or misshaping of the red blood cells, and basically what this does is it diminishes the oxygen carrying capacity of blood. So the red blood cells of individuals with disease contain hemoglobin S, which causes red blood cells to bend into this sickle shape. When it does this, it gives up oxygen to the interstitial fluid instead of bringing it to the tissues. Now, a little bit about sickle cell anemia. We'll look at our next slide and look at the shape of these sickle cells. So what does this mean? How did this develop? So this is a pretty interesting disease where we were introduced to a female mosquito, Anopheles albuminus, that had a protozoan, a parasite in it called Plasmodium vivax. Now, once Plasmodium vivax is introduced to the human body, it tends to beeline right to the liver, right to those hepatocytes, where it burrows its way in and takes advantage of hiding from our immune system to reproduce and reproduce and reproduce. Once those hepatocytes are filled with reproduced Plasmodium, the cells explode and then release Plasmodium back into the blood. So this is the second stage of onset of a malaria infection. So you first get bitten, it's introduced into your body, you get sick, right? You show the signs of your immune system responding to that parasite. Then it hides in your hepatocytes, in your liver cells, at which point your body thinks everything's good. Then it erupts and your body's like, infection again. So a second stage again of malaria. Now, what does it do? Now go back. I prepped you for this. Go back to those red blood cells. What are they? Sacks of protein, right? What's protein? Food. So Plasmodium found a way to burrow inside our red blood cells, hidden from our immune system, giving itself a ton of food. 300 million molecules of protein hiding in each sac, right? So we told you all that information for a reason here to lead up to this. So now what's it doing? It's eating and it's reproducing, eating and reproducing. Now we come to the part where those red blood cells are now going to be destroyed once it can't take any more of the plasmodium inside. And now we have the final onslaught. This is stage 3 malarial infection. This is usually what causes death. High fever. Your body cannot respond to this onslaught of parasites. And as a result, people typically die. Now, with malaria falciparum, we tend to see that it forms a sticky barrier towards that blood-brain barrier that prevents nutrients and oxygen from getting to the brain, again causing rigor and ultimately death. Now, where did this disease come in? Where did sickle cell anemia come in? Well, your body said, really enough's enough. I have to figure out a way to beat this thing innately, beat this parasite without really changing our anatomy. So what did it do? Our body invented over time this disruption, this disease, this imbalance, this sickled red blood cell. So now look, why did it do that? So again, there has to be a give and take. There's always balance in biology. What did we give up? Well, remember, a red blood cell lasts for 120 days. When you make the cell sickled, the body says, there's something dysfunctional with it, I have to kill it. So now every 5 to 7 days, it kills these dysfunctional red blood cells. So what did the body do? It essentially gave up that long red blood cell life cycle, that 120 days, which was malaria's, which was Plasmodium's third and final resting place right before its final onslaught. So it got rid of that last stage of malaria. So you could get bitten by a female mosquito, Anopheles albuminus, that's infected with Plasmodium, and have a malarial infection, but you'll just cycle through stages 1 and 2. You'll never reach stage 3. So this is a disease a disorder you would want, an imbalance you would want in areas that have high malarial infections, right? But now here's the downside. You have reduced oxygen or diminished oxygen carrying capacity of red blood cells, which means now you come to a place that is not overridden with malaria. We'll pick, say, anywhere in the United States and look at what we have. We have reduced oxygen carrying capacity, which means that high impact activities like running, sports, professional or even college level or high school level sports, these people with this disorder cannot really compete in because they run the risk of essentially dying, right? So they wouldn't be able to properly and adequately oxygenate their blood and subsequently their tissue. So pros and cons, again, balance in biology. Another good example of balance in biology, we're giving up this oxygen carrying capacity, but we're surviving a parasite that is predominant in or endemic to a region.